Hello, welcome to this lecture, Applied Animal Nutrition with a course code AES201. My name is ELISHA LYAWATWA, Tutorial Assistant in Animal Science at Sokoine University of Agriculture. Today we are going to cover the second session, which is the regulation of feed intake and theories of feeding. And after completing this session, it is my expectation that you will be able to describe the centers regulating the feed intake, but also you will be able to explain the theories that explains the feeding. And then after knowing these two things, you will be able to do the practical implication of the regulation of feed intake. So in regulation of feed intake, we have two major things that controls feed intake. We have the hormonal regulation, and we also have the NEFA's regulation. So what the hormonal regulation does and what the NEFA's regulation does in the feed intake, this is what we are going to discuss. So by starting with the hormonal regulation, you need to know there are two centers in the brain, and these centers are found in the very small part of the brain known as hypothalamus. So the hypothalamus is having two major centers. One of it is found in the lateral part of the hypothalamus known as the hunger center, but also we are having another part of the hypothalamus which is known as satiety center. And satiety center is found in the ventromedial part of the hypothalamus. Now, these two centers are controlled by two different hormones. And for the case of hunger center, the hunger center is controlled by the Ghrelin hormone. But for the case of satiety center, it is controlled by leptin hormone as the principal hormone, but also there are other hormones which controls the satiety center. And these two other hormones are cholecystokinin and the peptide Y. So these hormones are produced in different parts. For example, for the case of leptin, leptin is produced in the adipose tissue. It is produced by the adipocytes. So when the adipocytes are having higher storage of fats, it is when they start producing leptin. But when the adipocytes have low storage of fat, the production of leptin goes low. So you have to know that the leptin production goes directly proportional with the concentration or the amount of fats found in the adipocytes. But also there are cholecystokinin, as I said, and the peptide Y. The cholecystokinin and peptide Y are produced in the intestines of the animal. But also when it comes to the Ghrelin hormone, these Ghrelin hormones are produced in the walls of the stomach. And what happens until these hormones are produced is when the animal is feeling hungry, it means the stomach will be like condensed. It will be having the small size, not stretched. If the stomach is not stretched, those cells in the walls of the stomach will start producing the hormone called the Ghrelin hormone, and this hormone will go directly to the brain, specifically to the hunger center, to stimulate the animal start feeding. And when the leptin hormones are produced in those sites, as I have said, they will go also directly to the brain, specifically to the ventromedial part of the hypothalamus, which is satiety center, where it will trigger the animal to stop eating. So that is how this hormone works in order to control the so-called feed intake in animal. So after completing the hormonal regulation of feed intake, now I want to take you through the NEFA's control of the feed intake. We all know that the NEFA's control of the hormonal regulation of feed intake is always having slower rate. So the transfer of information through hormones from one part of the body to another is always at lower rate as compared to the NEFA's regulation. So the transfer of information from one part of the body to another through NEFA's system is very fast. So if the body could be just depending on hormonal regulation to regulate the feed intake, there could be some situations where the animal could not be performing well. So through the presence of some nerves, specifically vagus nerve, the one of the 12 cranial nerves, This nerve runs from the brain to the through the respiratory systems, the lungs, but it goes directly to the GIT. And in the GIT, this nerve has different afferent fibers that have touched different parts of the GIT. It is touching the stomach, but also it touches the intestines. So in those parts, this nerve transfer the informations to the brain. And you need to know one thing, that vagus nerve transfers the informations from the GIT to the brain, not from the brain to the GIT. So the transfer of informations when controlling the feed intake through vagus nerve is only one-way transfer, one-way system. The informations are running from the GIT to the brain to either trigger the animal take in the feed or to stop the animal from intaking the feed. So how is it working? We know there are afferent fibers that have touched the different parts of the GIT. When we start with the afferent fibers that have touched the stomach, when the animal has consumed enough of feed, it means the walls of the stomach will be stretched. And once the walls of the stomach have been stretched, that information will be transferred by the vagus nerve to the brain to tell or to command the animal to stop eating. Why? Because the stomach is full. The stomach is full. So the animal should eventually stop eating. But when the walls of the stomach have shrinked, meaning that there is no food in the stomach, the vagus nerve, through its afferent fibers, will also transfer the information to the brain to tell or to command the animal to start eating. Why? Because it tells that the stomach is having no food. So this is how it controls the feed intake when it comes to the part of the stomach, and it applies the same when it comes to the intestines. And this is what we call the mechanical transfer of informations through vagus nerve, because vagus nerve do two things. One is mechanical transfer of signals from the GIT, mechanical transfer of signals from the GIT to the brain, and the second is chemical transfer of the signals from the GIT to the brain. So with mechanical transfer, it means those mechanical activities, the stretching and the shrinkage of the GIT, it is what triggers those afferent fibers of the vagus nerve to transfer the information to the brain to tell the animal to start eating. Now, when it comes to the mechanical transfer or mechanical signals that transferred to the brain, this tells more about the cholecystokinin and the peptide double Y. The cholecystokinin, which is obviously produced in the GIT, when it is produced, there it goes and sensed by those afferent fibers that have touched the intestines. So when the afferent fibers have recognized the presence of cholecystokinin in the intestine, it tells the body that there is enough body reserve, and so the animal should stop eating. The animal should stop eating. And if there is less amount of cholecystokinin and less amount of peptide double Y, then the vagus nerve will transfer the signals to the brain to tell the animal that the animal should start eating, or to command the animal to start eating, because it tells there is deficiency of the nutrients in this body. So this is how the control of feed intake is being done. Remember, I have told you that the control of feed intake is done by two major systems or two major mechanisms. The first one is hormonal mechanism, and the second one is NEFA's mechanism. With hormonal mechanisms, you have to talk of the Ghrelin hormone, you have to talk of the leptin hormone, you have to talk of the cholecystokinin, and you have to talk of the peptide double Y, and you have to know the sites where these hormones are being produced. And when it comes to NEFA's control of feed intake, you have to talk of the vagus nerve, because this is the major nerve that has traveled from the brain to the GIT, and the major purpose of it traveling from brain to the GIT is to transfer those informations or those signals from the GIT to the brain, whether the animal should start eating or whether the animal should stop eating. So that is how we are talking of the control of feed intake. Now let's go to the theories of feeding. There are different theories which explains the feeding in animals, and these theories explains what we have already discussed with the regulation of feed intake when we talked about the hormonal, when we talked about the NEFA's regulation of feed intake. So these theories just explains how those mechanisms, how those control mechanisms works. Now there are like 6 theories which are much more applied. The first one is glucostatic theory. The second one is lipostatic theory. The third one is chemostatic theory. The fourth one is sensory appraisal. The fifth one is thermostatic theory, and the last one is gut feel theory. So today we are going to discuss these 6 theories, and I believe and I hope that by the end of this part, you will be able to explain these theories. How do they impact, how do they impact the regulation mechanisms of feed intake? Now, by starting with glucostatic theory, we say glucostatic theory. Here you just have to know about the presence of high level of glucose or low level of glucose in the bloodstream. When there is high level of glucose in the bloodstream, it means the animal has consumed enough amount of feed, and so the body will recognize, the glucoreceptors in the brain will recognize the presence of high level of glucose in the body, in the bloodstream. And so after recognizing that signals will trigger the animal to stop eating, because overeating will result into negative impacts to the body. But if there is low amount of glucose in the bloodstream, then this will tell the animal to keep on eating in order to increase this amount of glucose, so as to make the body in a neutral energy balance, not positive, neither positive nor negative energy balance. So this is how we talk about the glucostatic theory. And the most important thing which you have to understand is that glucostatic theory applies more in the non-ruminants. It is not applicable in ruminants. Why? Because in ruminants, whatever that you feed them, which is in form of carbohydrates, will be fermented in the rumen to produce the so-called volatile fatty acids, like acetic acids, like propionate and butyrate. So feeding the glucose, the glucose to the ruminants, or the carbohydrates to the ruminant, will always result into production of volatile fatty acids. So there is no there is nothing like glucostatic theory which applies in the ruminants. So if you talk of glucostatic theory, remember that we are talking of non-ruminants. So what is applicable in ruminant now? If glucostatic theory is not applicable in ruminant, what applies in ruminant is chemostatic theory. When you talk of chemostatic theory, we are talking of the level of blood metabolites. And when we talk of blood metabolites, we are talking of the things like ketone bodies, we are talking of the things like metabolites, volatile fatty acids, but also we are talking of the level of amino acids in the bloodstream. So in the ruminants, once you have fed them with carbohydrates diets, they will be fermented in that rumen by the lactic acid bacteria to produce those volatile fatty acids. So those volatile fatty acids will be absorbed directly by the rumen walls. They will be absorbed and when going to the bloodstream. So after fermentation and the production of volatile fatty acids, those volatile fatty acids will go to the bloodstream. And once they are increased in the bloodstream, it applies the same. The higher level of volatile fatty acids in the bloodstream will tell the animal to stop eating. The lower level of volatile fatty acids and other blood metabolites in the body or in the bloodstream will tell the animal to continue eating, so as to make the body in its homeostatic condition. So that is what we say about the chemostatic theory. Now, chemostatic theory, it is applicable in both ruminants and non-ruminants. It is not segregated in just one group of animals. And another theory is lipostatic theory. The lipostatic theory talks of the level of fats in the body of an animal. And remember, when I was telling you about the issue of regulation of feed intake, I was talking of, I talked about the leptin hormone. We say, with lipostatic theory, we say there are adipocytes, the cells which are found in the adipose tissue. These adipocytes are responsible in storage of body fats, the triglycerides. So when there is enough amount of triglycerides which have been stored in these adipocytes, then the adipocytes will start producing leptin hormone. Why are they producing leptin hormone? Because the higher level of fats in the adipocytes tells the body that there is enough amount of energy reserve, and so you have to stop eating or you have to reduce eating. So they start producing leptin hormone so as to control the feed intake, to control the amount of feed that the animal is supposed to eat. That is what we say about the lipostatic theory. And so with the lipostatic theory, the higher the overconditioning of the animal results into higher production of leptin hormone. When I say about overcondition, it means the animals which are much more fat, which have consumed a lot of feed and they have undergone fattening. They are highly fat, so they produce enough amount of leptin, and this leptin will negatively impact the feed intake. And the other theory which explains about the feed intake is sensory appraisal. The sensory appraisal just tells about the appearance of the feed, the color of the feed, the texture. So when the animal see the feed, remember in the previous session I told you about the appetite and the hunger. Animal can be satisfied, but because it has observed the feed which is much more apprising, which is much more attractive, then the animal will eventually start eating. So this will just, you have to talk about it in a way that if the feed is much more appealing, then the animal will eventually eat. If the feed is not highly appealing, then the animal will not eat. So that is how we say about the sensory appraisal. It is the same thing as how you are excited when you see the very cold Coca-Cola during the high temperature seasons, or when you see the tea during the very cold periods. You will eventually go for that because it will help you to either cool your body or heat your body. So that is what we talk about the sensory appraisal. And the last theory is the gut feel theory. With gut feel theory, we say when the rumen or the GIT is full of the feed, there are mechanical informations or mechanical signals which are taken from that GIT through the vagus nerve to the brain, and that will trigger the animal to stop eating. But if the GIT is having less amount of feed, then the information will be transferred to the brain to tell the animal to keep on eating. So those are the theories which explains the feed intake. Now, let's go to the last part of this session of today, where we discuss about the practical implication of this knowledge, the practical implication of regulation of feed intake. We say there are several practical implications, but here we are going to discuss only four practical implications. The first one is transition period in dairy cows. How is the regulation of feed intake operating in transition period in dairy cows? The transition period is the period when the animal is moving from, it is the period which covering the late gestation and the early lactation. The late gestation and the early lactation. We know during the late gestation or the last trimester, the animals always have low feed intake. And why there is low feed intake? Because the growing fetus in the womb causes the small or reduction in size of the GIT, or reduction in size of the reticulorumen. And because of that, the animal cannot consume enough amount of feed. And if the animal can't consume enough amount of feed, then this will result into negative energy balance. It will result into negative energy balance. And that negative energy balance will result into the mobilization of the body reserve. And what body reserve are we talking about? We are talking about the adipose tissues, the fats which have been stored. And the mobilization of these fats results into production of ketone bodies, And these ketone bodies then go back to the bloodstream and they suppress more the feed intake. So you find the animals which have moved from which are in the period of late gestation and early lactation are experiencing low feed intake because there is small size of GIT, but also there are high amount of blood metabolites which suppresses the feed intake. And because of that, you as a manager, you are supposed to make sure that you are providing the highly concentrated diet, a highly nutritious diet, so that even if the animal consumes less amount of it, but it will gain all the nutrients that is required. So that is for the case of the transition period in dairy cows. The second implication is overconditioned cows. The implication in overconditioned cows. How is this applies in overconditioned cows? We say overconditioned cows are those cows which have been highly fed, the cows which have been highly fed in their last trimester. The manager, maybe if the manager was thinking that feeding them at higher concentration will result into a good health of the cow, good health of the fetus in the GIT, will eventually result into a high accumulation of body fats. So the higher accumulation of body fats in the GIT, in the adipose tissues, will result into higher production of leptin hormone. This leptin hormone will suppress the feed intake. And remember, this animal, once it enters the lactation period, the level of leptin hormone will still remain high. And if the level of leptin is still high, the animal cannot eat enough amount of feed. And if it cannot eat enough amount of feed, and there is higher requirement of energy for production of milk, for maintenance, then the animal will have to go for the mobilization of body fats. And the mobilization of body fats will result into production of what we call the non-esterified fatty acids, or in short we call them NEFA. So these NEFA, they accumulate in higher concentration in the bloodstream. For the overconditioned cows, these NEFAs accumulate in high concentration in the bloodstream. And the fate of NEFA always, they go to the liver. And once they go to the liver, they undergo oxidation. So if they are in its standard concentration, they will undergo a complete oxidation. And the complete oxidation of NEFA will result into production of ATP, which is required by the animal now. But because this animal was overconditioned before, and there was high amount of leptin, and there was high amount of mobilization of those fats, which is by lipolysis, then there is higher production of NEFA. So the higher concentration of NEFA, when it goes to the liver, it overloads the liver, and the liver will fail to do the complete oxidation. And the result is incomplete oxidation. And what is the result of incomplete oxidation? It is the production of what we call the ketone bodies. And the ketone bodies now will go back to the bloodstream. Once they go back to the bloodstream, they will suppress the feed intake. But there is another pathway of these NEFA. Once they are still high in the blood and high in the liver, then what will happen is they will be re-esterified by the liver. The liver will do the so-called re-esterification of the NEFA to produce back the triglycerides. And these triglycerides are supposed to be taken away from the liver to the storage sites. And the thing which is responsible for taking these NEFA, these triglycerides from the liver to the storage sites are what we call the very low density lipoprotein. In short, we call them VLDL. So these very low density lipoproteins in ruminants is in low concentration as compared to non-ruminants. So if you have overconditioned your animal, and this has led to the production of triglycerides in the liver. Remember, there is less concentration of VLDL in the liver, which is supposed to do the exportation of the triglycerides from the liver to the adipose tissues. Now, because there will be poor exportation of VLDL from the liver to the adipose tissues, then those fats, those triglycerides which have been re-esterified will accumulate in the liver, and the result will be the fatty liver. Okay, will be the fatty liver. So the fat liver in combination with the ketone bodies accumulation in combination with leptin will result into the suppression of feed intake, and so the animal will be having no appetite of feeding, will have a low appetite of feeding. So this is very risky, and so you as a manager, you have to make sure that during the last trimester, you don't overfeed your animal so as to prevent the overconditioning, which is very harmful to the health of the animal post-calving. And the third implication is feed restriction and compensatory intake. Feed restriction and compensatory intake. During the growth of the animal, and for those who are doing what we call the fattening of the animal, they use this feed restriction principle. Feed restriction principle. So if the animal has been restricted, in amount of feed to take in amount of feed to eat, then there will be higher amount of ghrelin hormone in the bloodstream. But also there will be low concentration of leptin hormone in the blood. And so all these in combination will trigger the increase in feed intake. And because of the increase in feed intake, if this animal is exposed now back to plenty amount of feed, remember, if that feed is concentrate, then the animal will overfeed that. It will overfeed that. And the result of overfeeding the concentrate is acidosis. So you as a manager, you have to make sure that once you are taking your animal from feed restriction period back to the normal period, you have to make sure that the concentration of the composition of the feed is well balanced. The well balanced meaning that it have a standard amount of concentrate required, the roughage is required, so as to make sure that the animal is not undergoing the acidosis. And the last implication is the growth and finishing in beef cattle. How is the regulation of feed intake applied in growth and finishing of beef cattle? We say during the growth, always the animal are having less amount of adipose tissues, and because they have less amount of adipose tissues, then there will be less amount of leptin to be produced by the animal, and so the animal will be eating enough amount of feed, and that in continuity, and as the animal gets older, there will be deposition of fats. There will be deposition of fats in the adipose tissues, and that deposition of fats will result into production of the leptin now. And so the leptin hormone, which is being produced at the adult stage or at the finishing stage, this will be controlling the amount of feed to be intaken by the animal. And that is why we say the intake is always at its plateau state in finishing cattle, in finishing animals. It is not increasing, it is not decreasing, it is just in the plateau state, because there is this increase and decrease of the ghrelin hormone and the leptin hormone once there is higher amount of nutrients in the bloodstream, or there is less amount of nutrients in the bloodstream. And by saying so, this marks the end of this session. And I would like to welcome you in the next session, where we'll be discussing about the factors affecting the feed intake.
Chuo Kikuu cha Sokoine cha Kilimo SUA.